Hello everyone and good morning. I'm very excited to be here and to have all of you participate in our, actually we'll have two sessions, but this first session about research-based afforestation and dryland restoration. And just a few words before we start. So I think in the era of climate change and in the environmental crisis of desertification and habitat loss, it is clear and known more than ever that we have to base our land management on scientific knowledge. And today we wanted to show how KKL support and encourage such studies, environmental, ecological, long-term and short-term ecological studies in varieties of subjects to support our work. In this session you will see insights from the studies, from ecosystem to soil and tree dynamics. And I invite you all to listen and to participate and ask questions and to have an interesting debate after. And thank you. And for the first lecture, we have a special guest from Spain, Dr. Josu Alday. Josu is a senior researcher in the University of Lleida. Josu has worked in the restoration ecology of Mediterranean ecosystem in Spain, mainly in mine site and forest ecosystem after wildfire. He has been focused recovering plant cover and the plant soil feedbacks in degraded ecosystems. So, please.
Thank you very much for inviting me. It's a pleasure to be here. Well, I feel like a singer star. It's a pleasure to be here, and I will give a conceptual talk about one of the things that usually we don't consider when we start working in restoration or recovering some ecosystems. This is normally when we work in recovering ecosystem, we focus mainly in the plant side or the plant or the cover of vegetation and all the different elements, but we lost one important element of this ecosystem, which are the plant-soil interactions, and some of them related with the fungi. This doesn't work. Okay. I came from Spain. Spain is a Mediterranean country mainly. We have a long history of mining activities in Spain. This is the Cartagena-La Unión mining district in the southern part of Spain, quite clear Mediterranean climate with a low precipitation. And this mine area has been open, has been in constant use from the Roman Empire. The Romans were the first ones to come here to dig for silver, and nowadays there are some small exploitations, small mine sites. But the landscape has a very big problem because we need to recover a lot of hectares of impact created by humans. This is how they look. Some of these mines that were abandoned in the middle of the 20th century. You can see that there is not recovery in vegetation. There is nothing to establish there. So we need to work hard. One of the things that happens when any restoration project starts, or the people think when reading the news that we are going to start recovering or restoring this mine site, or the general idea that the people has, or the local authorities, is that when we are going to recover these mine sites, we are going to be able to create these things, these kind of ecosystems. It's funny, but most of the people think that, well, these guys are going to come to here to recover the forest ecosystem. They are going to create this kind of Amazonian ecosystem, or even the forest that we can find in Connecticut, for example, right? So this is not possible at the end in our Mediterranean landscape. Well, it's possible, probably. All the things are possible, but we need to invest a huge amount of money to do these kind of things. At the end, there are a lot of goals of recovering, goals of what people do, what people want, a lot of things. And one of the things that is funny is that when we try to recover this dryland ecosystem, there are two different elements or two restoration ideas that are important. Is that from a point of view, we need to recover ecosystem functions, which are important to keep the ecosystem alive, but also for the human well-being. Also, we need to recover some underground components. And this is important because the underground components are those which are creating the carbon sequestration, the nutrient cycling, which are part of the healthy function of any ecosystem. But in most of these restoration programs, we are missing one important element, is the plant-soil interactions or plant-soil feedbacks. We are only focusing maybe in the easier part, which is the planting things, planting elements, but we are missing this plant or this soil biological component. And especially one thing that in which I worked from the last year is that relationship with trees. All of us know that the trees need some kind of symbiotic relationship with fungi to get some nutrients from the soil. So there are some mycorrhizal species that relate with trees, and most of the trees that we know, they got this relationship with this mycorrhizal species. Okay, so we can find two different kind of fungi in this kind of ecosystem. The saprotroph, we use the organic matter, and the ectomycorrhizal species that are closely related with the trees, that they help them to get nitrogen, for example, or different nutrients from the soil. Okay, this is the theory that we know from ecology that when we learn from our university degrees. Okay, but these concepts are not usually applied when we try to recover ecosystems. Also, recent research shows that these different fungi species, saprotrophs and mycorrhizal, are very important in nitrogen cycling, for example, on the carbon cycling in the ground, and they are responsible in the carbon fixation and also in the nitrogen cycles. These cycles are really important for ecosystem and also for trees because they help them to grow and to establish in a better way. And it's funny because it's saprotrophs. All the people thought that saprotrophs decompose organic matter and also absorb some carbon from there. But also we have seen nowadays that there are mycorrhizal fungi that can not maybe decompose, but through some kind of oxidation they can get the nitrogen from this organic matter and prepare and transfer it to the trees. So at the end, having organic matter in the ground or in the soil is important for a healthy and established some forest ecosystems. But this is the theory behind how the fungi relate with trees and what is the importance of fungi in trees. But what we do when we face to a recovering one-man site, as we saw you before, we do this. We go there and we plant a tree. Okay, it's a good option. It's cheap, it's easy, all right? But it is. We are planting a tree. And what we expect planting this tree? We expect that with time, this tree will evolve the root systems. They are going to relate with different elements of the environment, of the ecosystem. All the species that are going to arrive here, fungal species that are going to arrive, they are going to be here. They're going to find each other. The fungi will find the tree. They are going to make this kind of funny relationship. I will like your carbohydrate. The tree is going to be, I will like your nutrients. And all this happy ending will happen. We are thinking in this. Okay. But the truth is that we are planting a tree. Most of these trees came from greenhouses. The fungi that live in greenhouses, maybe they are not adapted to the ecosystem. We are going to go there, and in the new soil, what's usually happens is this, is the Game of Thrones. I mean, if someone came here, and who is this guy? And he's not well adapted, right? So this creates some handicaps to see, well, are we doing the things correctly? Maybe from the vegetation perspective, it's okay, but maybe considering all the ecosystem in a world, considering the soil, plant feedback interaction, we are maybe not doing in a proper way. And especially if we consider that in trees or in the plants' life stages or in the different plants types, there are different types of fungi that they are related with this kind of species. And this is important to have in mind, because maybe we are planting something in an ecosystem which is not, especially not well prepared. Maybe we are introducing a different soil community of fungi, or maybe the species is not adapted properly to the communities that are there. So we will have some problems at the long term. This is the background, and this is the idea that I usually show to the people, and here comes the scientific part of this thing. It's normally when restoration ecology started 20 or 30 years ago actively, there were no tools to analyze the soil or the bacterial in the way that we are doing nowadays with DNA analysis techniques and that things. So this could reduce or hamper the possibility to consider all the ecosystem compartments when we were doing restorations. Okay, so I think we wanted to test if we were doing correctly, not from the vegetation perspective, but also from the soil community perspective. And we started to work in our mine sites here in the Catalonia region, which is Mediterranean Spain. And we wanted to see if different restored areas were related with different fungal communities that were in the environment, or we were creating new communities in these ecosystems, or what was happening there. Especially, we wanted to know if we were recovering the ecosystem functions, and if we were recovering the property functions that we think that each of these restorations should have in its time. For that, we use, as I said, we were collecting soil samples. We use new DNA techniques to analyze the DNA of these fungi and try to identify the composition, but also to try to identify some functional genes related with the, for example, with nitrogen. And we analyze so we make some bioinformatic analysis after that, and finally, we were able to get the compositional matrix to identify these fungi, how they change the composition of these fungi, how they change, what were the processes that they were doing, what were the relationships, but also we were analyzing which bacteria were related with these fungi or different things. One of the funny things that we found, or one of the clear things that we found, is that when we were recovering some different abandoned mines or different mines in different sites, we see that there were, depending on what kind of ecosystem was the target of these mines, each of them, they have different nutrient cycling, and especially when they were dominated by different nitrogen cycling processes. For example, in abandoned mines recently trying to recover to a grassland, there were a lot of nitrogen fixation, and there was nitrification in Juniperus turifera and some calcareous grassland with very high interest in the region. But there were some denitrification when we were moving to more Quercus-dominated vegetation, like Quercus pyrenaica, which is an endemic species from the Spain, Portugal, and Morocco. And when we changed, especially from Juniperus to Quercus pyrenaica. So this gives us an idea that maybe when we were analyzing we wanted to change the vegetation from one side to another, also we need to consider that we also need to change the cycles that are related with this vegetation, or almost to be able to identify what the cycles that we are creating in the new ecosystem match the cycles that can be found in different areas. And also, it's from our side was interesting to identify in the forest vegetation areas if there were a lot of differences in the species that we were able to find in the fungi communities, and we found really that there were not higher differences between one side or another. I will expect this because they were close sites, And and usually fungi, like bacteria, they got a very extremely high colonization capacities. And if they are, if the plants that we are placing in one side have similar plants around, they can be able to colonize this ecosystem quite easily. One of the problems that we said is that when we were including, for example, pine species in different restoration approaches, what we realized that what we were doing from a soil community composition perspective, we were homogenizing all the communities. And this is one of the interesting things, because it's even in quite far ecosystems, when we were planting pines, we were creating almost the same communities from a fungal perspective. So what we were doing was kind of homogenization the fungal community and reducing the spatial diversity of one of these restored ecosystem. And this is one of the things that we need to consider in the future, because it's fungals represent a huge amount of biodiversity in our ecosystems, for example, in the Mediterranean ecosystem, because in 1 gram of soil we can find them from 2,000 to 3,000 species, right? So if we create this kind of homogenization of communities, we are altering in somehow the biodiversity of the restored areas that we are creating. Maybe not from the vegetation perspective, but the biodiversity as a whole, considering all the compartments. So, what was our idea to recover and to try to include the restoration programs, these soil communities? Well, it's difficult because we don't have a lot of knowledge, and as I told you before, trying to create a new community in some ecosystem is difficult, because there in the ground is going to be a Game of Thrones playing, and the results are not very successful nowadays. But one easy recommendation that we apply nowadays in our research areas is that if you wanted to recover some marine ecosystems of some Mediterranean areas to pine species, it's easier to use some kind of inoculum from the previous pine area, and this could be, for example, later, to try to give to the tree sapling the opportunity to have the surrounding area inoculum of the mycelia, to try to give them some amount of fungi that can be relate with them, and try to find the best opportunity with this fungi. And if we wanted to recover oak species in our mine areas, it works slightly different, because we need to use normally humus or soil layer, and not the litter layer, because the litter layer in oak species is mainly composed by saprotrophs, instead of in pine species, which are a combination between saprotroph and ectomycorrhizal. So in oak, if we want to recover oak, we need to cut some kind of inoculum from the soil layer of the natural of this ecosystem, to try to place with the saplings, to help them to establish or to give them the possibility to choose the selected fungal communities. This is from theoretical perspective and some studies that we have developed. We don't know how this works in a landscape scale, or we don't know exactly how this works in a medium and long term. It could be problematic, it's true, but I think this is a new path to try to work in recovering, and also understanding how the plant shade feedback and how the different elements of the ecosystem can be restored at the same time that we recover in the vegetation, which is the most important part. So with this, I will finish with 3 conclusions quite related with what I told, that it seems that we are creating different soil fungal communities with different functioning when we are recovering some ecosystems. This could not be a problem at the beginning, but maybe at the future we are going to reduce the amount of different communities of fungi that we are going to be able to find in our areas. Also, I will say that we will place more focus on soil biotic component in restoration programs, and this is important. I know it's difficult, now we are start to having the tools, but the soil biotic component can help to improve the restoration process that we are doing. Only including in the programs and thinking about it, it will help us to do more suited restoration activities. And from restoration application perspective, including fungal communities and plantation programs could improve probably the chances to recover in these communities and some soil functions. Okay. Thank you very much, and that's all. I think I fit to the schedule.
Thank you very much for the talk. This was really important. Can you go back to the slide with the abundances?
Yeah.
This one. We're looking at the ectomycorrhiza on top and the avuscular on bottom? Or?
I think those are saprotrophs, probably. Yes, saprotrophs.
And these are mycorrhizal.
And those are mycorrhizal, yes. This is an oak forest, and this is revegetation playing with oak forest in some kind of. So they are quite similar in somehow, but well, this is to show that if we do it in a proper way, we can have some similar results in the way.
Revegetation, you mean the planting?
Revegetated, this is a funny thing. Revegetated plain was theoretically planted, but I don't think it was theoretically planted. I think it was not planted. Okay, so most of this plain area were recovered. I don't know why. I will expect that natural regeneration or natural recovering of the trees that they were there. Okay. Most of these differences are produced because those saprotrophs behave different in this oak forest than in the revegetated, which are dominated with shrubs and also with some other species.
Could you explain what are the key factors that affect that abundance?
The key factors that affect abundance of the saprotroph species, for example. Well, good question. Well, if we consider the soil conditions are very important element to restrict like normally, but I think there are two different elements that restrict the abundance of some of these species. First of all is the organic matter in the soil, okay, and the other one is the water. Okay, this is two elements that are related. In a very dry time in Spain, we can find a biomass of very low biomass of some of these mycorrhizal, and when we sample the same in October or November, we will be able to find more biomass. And also there are differences if we are analyzing the pine species and the oak species. The behavior of the ectomycorrhizal species in the pines are different than in the oak species. In our ecosystem, in oak species, the ectomycorrhizal are trying to protect the root systems, and they are mainly in the soil, and they are protecting them from the different pathogens and helping them to absorb some nutrients from the ground. But in pine species, the most of this ectomycorrhizal layer is in between the leader and the first centimeter of the soil, because there is a kind of a fight between the ectos and saprotrophs to obtain the nitrogen, because the pine trees in our ecosystem, they need a lot of nitrogen to grow fast and try to produce a lot of seeds. Okay.
I wonder if you have any insights on the first stage of coming from the first video you showed us, the first picture of the mine.
Yeah.
It's a very degraded land, and I think it's representative for also a lot of the area of the northern Negev. And how do you do this shift to restart the restoration? So I think the early stage is a question, maybe if you consider soil, so maybe some practices before planting.
I would say something that maybe will sound weird here, right? But here in this picture, I will never do nothing. I won't do nothing, any restoration activity. And I will say why. Because I think that this is a cultural landscape from Spain. Nowadays, there is an idea to try to recover it, to try to plant and to try to do a restoration of this area. From my point of view, leaving it this small area as it is can show the people of future generation how these all mining activities they were done, because this was mainly dig by hand in the 19th century until the middle of the 20th century. Okay. Responding to your question, here we have a problem with pollutants. There are huge metal pollutants in the ground. So first of all, we try to cover them, try to provide some kind of normal soil layer to try to establish elements over it. The thing is that there are steep slopes. This is a problem because when we get rains here, they are normally pouring rains. There is not raining normally, and most of these areas run outside. I think if we wanted to recover ecosystem in this kind of areas, the first step is to provide some stable soil with organic matter. This should be fundamental. After that, we can establish and plant different things. But establishing a good soil will increase the chances to have a good restoration in this ecosystem. This should be our best. And also, this is why I place this picture. If you see the side vegetation which is original, you will realize that there is not a very dense stand of forest. Okay, we can find some species adapted to the Mediterranean area, some shrub species, some olives. So we need to create this kind of ecosystem resembling the natural ones. We don't need to create a big stands with eucalyptus or with big stands with pine species, with very dense ones. All right. So this is the target or the idea that we need to provide.
Thank you.
Okay.
So for the next lecture, I would like to introduce Dr. Eli Argaman. Eli is a senior researcher and the former head of the Soil Erosion Research Station at the Ministry of Agriculture and Rural Development of Israel. His expertise and research interests include agricultural knowledge management, implementation of sustainable land management methodologies, soil erosion and preservation, detection of land degradation processes, and remote sensing. And also I added that he's a leading researcher for one of the KKL's long-term ecological research station located in Shagririm Forest at the northern Israeli Negev.
Okay. Well, this presentation is supported by the JNF, which fund all the research in the Ambassador Forest area. And my talk will focus on the general long-term findings that we are working on starting 2008, but we are monitoring more than 20 years in the specific location, and I will expand very soon. Land degradation processes, some people might call them natural or anthropogenic, affect the functioning of dryland systems. These processes are gradual and mostly in the short term undetectable. And the possible result if the ecosystem collapse is mostly sudden, because until you see it, it takes sometimes a year or two, but after a decade the collapse is happening in a very short time. One of the major drivers of land degradation in our area is overgrazing, particularly in other semi-arid areas as well, but mostly in Israel. This is one important driving factor. This affects the acceleration of the land degradation and significantly affect also the soil ecosystems. For decades, afforestation projects are widely accepted as an effective and important means for mitigating land degradation processes and also adaptation to climate change. And this is also common in our region. You can see some pictures on the slides, but showing the view of these areas. Across Israel, especially in the semi-arid region, afforestation projects are common and implemented by the JNF and aim to restore degraded lands. Most of these restoration projects rely on water harvesting systems that you can see on the picture over here, which are focusing on two major practices. The first is dike terraces, which are constructed along slopes, and limans that are constructed along gullies. This system have been established in hill slope and different valleys, and they support woody vegetation like trees and shrubs in addition to grassland development. The long-term objectives of this project and the specific area is to assess the temporal dynamics of land use change following water harvesting construction across a large-scale dryland afforestation project. The area, the Ambassador Falls, as you can see over here, is in the white polygon, and we're focused on 3 specific locations with 3 different planting ages. The first one, in the blue polygon, is the area that was planted during 2005, after the construction of the water harvesting system. The second is a planted area that was constructed in 2009, and the most new is in 2016 area. So we have 3 polygons across the whole forest location. And for this comparison, we also compared the locations that we are monitoring in ground, also in aerial and satellite, and we have some control locations that are slopes but without any water harvesting construction. The yellow points that you see are the MODIS points, pixels that we are monitoring in a specific location starting 2000 until date. And they are exactly in the location that we are also performing ground truth validation. So the area of the forest is extended over about 25 square kilometers and over moderate slopes and an angle of 3 to 9%. The hill slope afforestation system, well, people call this a forest, but the canopy coverage is very, very small. It varies from 0.1% up to 1% of planted trees. A month ago, I gave a presentation describing this project in Italy, and they said, where is the forest? Well, this is the forest, and you can see the density of the trees. And before the establishment of the project of the forest, Ambassador Forest, the area had been really exposed to a very heavy grazing for many, many years. And this accelerated severe degradation processes that really had a meaningful impact on gully formation and even some hazard to infrastructures. And following one of the rules, following the plantation of the vegetation, the woody vegetation in the water harvesting slopes, is that it's prohibited to graze for at least 5 years, in order to give the system some time gap to adjust. Some background about the climate in this area. So, well, everybody knows that we are facing climate change, and where you can see the green dashed line, this is the long-term average rainfall by the Israeli Meteorological Service from 1990 to 2020. The average for this period is about 290 millimeters per year. Our monitoring date from 2000 to 2020, we found that we have a lower value of annual precipitation, which is about 260 millimeters per year, and you can see this on a black dashed line. During this monitoring period, we faced a very long drought. Started from 2005 and ended in the end of the year, hydrological year of 2012. Hydrological year starts in the 1st of September and ends on the 31st of August the next year. So we had a series of drought years, and they are really visible on the atmospheric database. And what we see also, it have a significant effect on the rain days amount and rainfall amount along the season. The season starts from September, as I mentioned, and in August. And we can see that during the drought years, a significant reduction of number of rain days has appeared, and also a significant reduction of rainfall. And we can see also there is a big shift on the amount of rainfall. When most in a normal year, most of the rainfall falls in about January. During the drought years, the highest amount of rainfall appeared in February. Now, what we see here, this is the timeline distribution of the MODIS NDVI product of the Terra and the Aqua satellites. And we see here the deviation between the three planting areas and the control, unconstructed slopes. And the upper line, blue and red, are the NDVI fluctuations along the years from 2000 until the end of the year of 2020. And we can see that the gray shaded points are the planting years of each location for 2005 in that area and 2009. This position and 16, which was beyond the drought year, series of drought years. The lower graph shows the difference of the annual the monthly NDVI comparing to the long-term NDVI. And we can very easily see that during the drought years on all locations that the rate was negative comparing to the long-term average of the NDVI. Now, this is a deviation between the years that were not considered as drought and the 5-6 years that were considered as drought years. But if we look on a different aspect, then when we divide the time period before the construction of the water harvesting system and after, the blue is before and the red is after the construction, we see a different trend in each location, where the most meaningful increase from the construction in 2005 is a 0.4. The correlation is high and significant, and also in 2009, but was actually constructed in the middle of the drought years. And if you look on the youngest location, which was planted in 2016, also a dry year, we see that there is no significant trend before and after the construction of the water harvesting system. And the behavior is equal or even better comparing to the unconstructed slopes. If we look on the immediate impact of the construction on each location, planting location, we see that it's actually visible very significantly in each planting location, 2005 planting area, 2009, 2016. And what is the most interesting, that each one of these years of planting was a dry year, also in 2016. And we see here, this is an example, but even during a dry year, the planted locations are behaving much better comparing to the location that was disturbed in this specific year. So it takes time to recover, but the immediate impact is visible. A very interesting finding is that what we see in this slide is that the regulation of the phenological phase of the vegetation during the annual, during the rainy season, the hydrological season, is regulated after the construction of the water harvesting system. We can see this in the location of planted in 2005 and 2009, but we see that the behavior along the season becomes very regulated, and we have a nice distribution before the peak, which was present before the construction, and even in 2009, the plantation area, there was a lower, vegetation coverage and density. We have an increase following the construction of the water harvesting system, and the most significant effect is that it's actually a result of the annual vegetation, not the trees. Remember, trees are 0.1 to 1% coverage. We are talking about the annuals, which have the most significant impact on the surface coverage and density. The area that is not balanced yet is the youngest one in 2016, where we see a reduction, but the distribution before the construction and after is completely different, and it's not balanced yet. This all is compared to the control area, which is actually almost identical to the upper two locations. So if I will summarize, we see that the establishment of the water harvesting system has a very adverse short-term effect on the herbaceous vegetation cover. The immediate effect is sharp and appears in reduction of the annual median DVI during the year of the construction. In the long term, self-restoration and geo-ecosystem prevails, suggesting that mitigation of runoff water loss from the hill slope and keeping it along the slope is a very positive parameter. These findings actually underline previous studies that we made in this area, and that hypothesized that in the semi-arid region, ecological self-restoration is expected to be affected, positively affected about a decade after the establishment of these harvesting systems. Thank you. A bit louder.
This water that's being harvested was supposed to actually be somewhere else, right? I mean, if it wasn't...
Yeah. If you will not harvest them, they will end on the stream or a gully, and will destroy the gully and will have a severe impact on it.
So it has a positive effect on the downstream?
Not necessarily, because first of all, I'm monitoring this area physically since 2008. Runoff effects on downslope are almost invisible, are not present almost none of the years. And the effect is that you are mitigating the runoff, and you're not affecting downslope because the amount of runoff have a much more severe impacts, infrastructure impacts on the ecosystem, on the stream ecosystem, comparing to the mitigation, because we're not completely stopping the runoff, we are mitigating the peak. So it takes longer time to flow, but in the same time it reduces erosion and damages to the slope and the soil. There's another presentation I have the impact on soil quality, which is really visible and can see very the significance of the positive effect of this construction. Louder, I can hear you.
You observe the vegetative cover and you will evaluate.
I'm just a plain soil scientist. I don't deal with that. Tamir? No, we haven't checked this. This is now under study. I dealt only with soil properties and the water harvesting system in this project. We are now studying this impact of the diversity, species, but I'm just a plain soil conservation scientist. I know what is green or not.
So our next lecture of Dr. Eli Zaady. Eli is a soil microbial ecologist, senior researcher in Natural Resources Department of Gilat Research Center, Agricultural Research Organization, the Ministry of Agriculture of Israel. He specializes in research in open landscape of dry environment, soil, vegetation, and biogeochemistry. Sorry.
The system that I want to describe today is the Sayeret Shaked. Where's the arrow? Oh, okay. Sayeret Shaked area. I worked in the northern Negev. Over here, it's a semi-arid desert with a long-term annual average of 200 millimeters of rainfall. The LTR is actually devoted to certification and restoration and climate change research. And I put this photo from fenced area from the end of the 80s. You can see clearly that it's a patchy area over here, just patches of shrubs, and you see biological soil crust, which I call it a biocrust over here between them. And if we're looking at the small scale, what is happening from the biocrust hydrologic function, you can see that the crust is actually produced from bare soil, and you can see more soil mound under the shrubs over here. And of course, rainfall and the biocrust in this area producing runoff water that increase the soil moisture and the water conservation. However, you can see that not all the water is actually getting into the shrubs. And I think I have a better one over here. The water that's coming from the biocrust are catched by the shrubs over here, or the soil mound, but some of it over here, it's fenced, so we know exactly how much water is coming out, but some of the water is leaking out from the system. If we're looking from a hill slope scale, thanks Eli for some of the data over here, if we're looking at the runoff over here and the patchiness between the shrubs and the crust, you can see that as we have more shrubs, the runoff decrease. But still, some of the water running out. So the idea was very good and was used by the Keren Kayemeth LeIsrael, is to build a soil mound, 1 meter soil mound, and to catch the water, the extra water that's running out, usually from the system, to plant trees. This was a very good idea, and if you're looking at the overland runoff production, in average rainfall amount, and you can see the runoff of that undisturbed area, and from water with the counterbanks that was built by the JNF, Keren Kayemeth LeIsrael. And the area is looking great. It was looking great. In 200 millimeters of long-term annual average, you can see the savanization project, trees, green trees, and you can see the soil mound and the biocrust in between, so everything was looking great. However, and this, thank you, Eli, for all the previous explanation about what's happening over here. Look what's over here. We have a drought over here, but then the rain increased during the year later, and it was, let's say, have some kind of water back to the system. But if you're looking over here, the rainfall, although it increased, it's not increased to the level before, and we call these years very drought years, and the rainfall average dropped from the 200 to average about 150 millimeter of rainfall. Nothing happened. Okay. So what's happened over here? We have the biocrest that's producing runoff water. I think the battery is lost. Finished. No. Oh, okay, good. What do you do? First one? Okay. So what was happening over here, that the biocrest produced runoff water, and it's not helping. Okay, I'll give it up. And some of the water intercepted by the shrubs, and we have the soil moisture, and everything is looking nice. If we are calculating the amount of in 200 millimeter of rainfall, this was before the drought, okay, we have 1 millimeter rainfall for 1 cubic or square meter is about 1 liter. So the crest runoff harvest area, that amount producing the runoff, and we have 140 square meter. That's where we calculate 10 meter between the 2 counterbanks and 7 meter between the trees, in average, of course. So the calculation was telling us that we have 200 millimeter of rainfall, and this is the average percent of the runoff that's produced by the biocrest, multiplied by 140, and we get about 5,320 liters from the contributing crested area. And we have 2 more meters over here that's usually plowed in order to increase the infiltration. So the total is about 8,120 liters in 200 millimeters of rainfall. And this is what's happened to the shrubs in this area. We have beautiful shrubs. This is stage 1. Look at I cannot see the year from here, until 2008, and he's from 2008 to 2012, 13 to 15, and 2015 to 2020. And you can see what's happening to the shrubs, dry out, and this is the soil mound that we can see and what's left of it. What we have now is we say it's okay. The area was looking like that because most of the shrub died out. So we're going to have more water, and it's look great. Okay, it's getting. We have a lot of runoff water. Of course, the shrubs are dead, most of them, and we have a lot of rain or runoff water running over there. And it was good photo, but I need to stand in the rainfall and take this photo, so it was not so nice. Okay, but let's calculate what's happened with 150 millimeter of rainfall. Now we have about 4,000 liters, and we will add these 2 meters, so it decreased from 8,120 to about 6,000 liter, a decrease of about 30% of the rainfall in this area. So we wanted to see. We went to the area, and this is the aerial photo of Seyeret Shaked Park LTR, and this is the fenced area, and we choose some. This is the area that Eli is working over there. and we choose 4 plots. This is area that's all this area, of course, not all the fence, but all the area are grazed by sheep for Bedouin herds, and we choose a plot over here, 1, 2, 3, and 4, and these are the list of the trees that were planted over there. The blue one are I mark with those that we have wide leaves trees. And the difference between the 3 and the 4 is that the 3 was in the beginning, after planting, before planting actually, had a tillage that destroyed all the crust, and this area is not used for water harvesting technique with counterbanks. Plot 1, everything look great. Okay, we have the Prosopis alba. We don't feel, the shrubs are dying, but the tree is looking great and everything looks nice. Here you see the dike over here. It's very nice to see that. Plot number 2, look what's happened to the eucalyptus. This is before and this is after. In the hill slope scale, a lot of them dry out. Plot number 4, that's what left. There was a lot of trees over here and not much left. Okay, this plot with the counterbanks, you can see that some of them still green in the crested area. Crested area with the water harvesting technique that Keren and LeIsrael are using. So we compare between plot 1 and 2. Just remember, this is without counterbank, this is with tillage, of course, and this is with the counterbank, and you see that, and here we put trees and their drought tolerance, and we mark the low, the medium, and the high tolerance, and you see that these three, the ficus, the schimus, and the Ceratonia just vanished. The white leaves vanished, and we cannot see them anymore. Those that we said it's a medium tolerance, you see that the percent of survival dropped to 27. Of course, we did it from the trees that were planted in the beginning, and over here it's about 51. The pistachia dropped over here to 6.5 and 27.1. And what was interesting to see over here, and we don't know if it's replanted or not, but we saw increase with the Acacia radiana, which is local trees in this area. And the Prosopis have a good survival too. Oops, sorry. So these are the trees and how they look like. This is not my photo, so I took it from the website. So the long-term research in Park Sayre-Shaked made it possible to study the ecosystem response to a higher frequency and magnitude drought as a consequence of climate change. The studies before and after the drought induced system collapsing, made it possible to quantify the effect of climate change on the planted trees and their tolerance, the role of the biocrust as environmental engineers that regulate the runoff in this area, and the water harvesting techniques helps to tree survival following the drought. Thank you.
Hi, thank you for the talk. Can you elaborate a bit on why, for the high resistance trees in drought, there is an increase in survival in the entrusted system rather than in the entrusted one, while in the mild resistance crops it is the opposite?
Let's go back to the numbers. Over here, you see, that's what you're asking, why there's an increase in compared to this one. The Prosopis are not a local one. It came from, I think, from Mexico. And why it's increased over here? Well,
Regarding the influence of the crested over the extra,
yeah,
the crested providing more runoff.
exactly. So maybe it helped, but we don't know why it's. Are they sprouting themselves or whatever? We don't know. the birds are spreading the seeds, or this is a averages number. We don't know what's happening in this area, but what's important that you see the levels of between this tolerance, and probably not to plant these trees again in this area, or if we in the future we're going to have more drought, it's going to be a big problem. But to plant only acacia radiana in this area, it's not so nice. So we need to look for other trees.
Yeah, so apparently we have Zoom and everybody needs to hear me, so I need to practice the height of the microphone. The next lecture will be given by Dr. Tamir Klein, and I think you already saw the shift from soil dynamics. Now we had Eli with the tree dynamics, and Tamir will give more of an ecosystem perspective. Tamir is a tree ecophysiologist, and since 2016 he is the head of the Weizmann Tree Lab. Before that, Tamir was a PI at the Volcanic Center of the Agriculture Research Organization, and he did his postdoc in the University of Basel in Switzerland. Please, Tamir.
Okay, hi everybody. So I'm not Ella Posner, but I speak on her behalf. She was a master's student in my team, graduated. She's now a data scientist in a high-tech company, so you will see a lot of data in this talk. So we stay in the same neighborhood from Eli Zaady and Eli Argaman, but we go just a bit higher in elevation, a bit cooler, and just a bit more precipitation. So 50 kilometers north from here, if you go straight, you hit what we call the dry timberline, the end of the forest. And here I mean really something that really looks like a forest even to European eyes. Like almost complete canopy. This is the Yatir forest. And if you go and visit, and I hope you do, you will see trees flourish there. By now they are more than 60 years old, and a lot of mature adult trees. But if you have a forester's eye, you see something missing, and you don't see the next generation of the forest, and this is something missing, and we wanted to understand why and what happens to seedlings and how it happens. So that was the work of Ella, and that's what I'm going to show you now. And that's what we think about as a hidden mechanism of forest loss, because even that you don't see trees dying, you can have a forest that will just die from old age after 100 years, and no other trees to continue that forest. Okay, so forest sustainability is relied on the parallel process of adequate growth of mature trees and the ability to recruit the next generation. This is what I've just explained to you now. Forest disturbances turn extirpation. This is like local extinction of species. And here we don't talk about acacia or eucalyptus, but Pinus alepensis, Aleppo pine. We have the friend from Spain, so this is a major tree of Spain, I know. It's a pan-Mediterranean. So in the Mediterranean region, it's the most important tree species. And when we talk about seedling recruitment, so let's go down and see what happens to these young, very young seedlings. They have to germinate, of course, and then they have to survive the conditions outside. And in the end, if they survive more than that, they grow like a multi-year growth, and then we can measure how they grow and so on. So seed germination happens in this forest in April. Seedling survivorship we measured in September, just after the dry season. So these are 6 months old seedlings by then. And multi-year, we measured again in September. And on the far right corner, you can see the monthly precipitation. So it adds up to 280 millimeter per year on average, but there's a big variation to that. So this is how the forest looks like. You see some agricultural land in patches, but the forest is mostly an Aleppo pine forest in the hills of the Judea. This is not very far from the wild range of Pinus alepensis, just 10 kilometers south of where it should have been. Okay, and it's I don't know, massive, but it's a big man-made plantation. And it is located here, as you can see on the map. And here are the variations between years. pay attention, it's starting with 100 millimeter, not 0, but still very big variations. And as you saw before, we had some subsequent drought years that affected the many ecosystems, the forest included. Okay, so the hypothesis was, if Yatir forest is a marginal tree population on the dry timberline, that seedling recruitment would be lower than needed for long-term forest regeneration, which in turn can jeopardize the forest's future. And we also tested, as the other stations that you saw here today, which are long-term ecological research stations. That's a unique program in Israel. It's part of the European program, a global program, that we are in place for decades. And we measure always the same things in a very structured manner. So we created an experiment with two major forest management activities. One controlling the density of the plots, and the other the grazing. So by density we mean, I will show you immediately, 3 levels, either 100, 200, or 300 trees per hectare. And by grazing, we know that grazing can reduce fire frequency and competition with other plants, but can also involve injury to young seedlings. And in the Yatir forest, grazers feed on already sparse understory vegetation. I want to share with you something, because many people don't understand what we mean by grazing. So maybe I try to show this. I don't know if it works. Maybe not. Lamata? Okay. Let's see if it works. That's not what I meant. Okay. Later I can show you when you come to me in person. But this is not bacteria. This is sheep from a drone. And when a herd of sheep and goat like that goes into a field, it's amazing what it can leave behind, which is not much plant. Okay. So we hypothesized that, where did I go? Sorry. That thinning and grazing exclusion could improve seedling recruitment by providing higher light and water availability to seedlings while preventing grazers' damage. And the first hypothesis was the seedling recruitment would be lower than needed. That's what I said before. And this is a big experiment in the field with more than 20 plots. You see the fences in each of them for keeping the sheep out. And you see the different densities by colors. We will stick to these colors for the 100, 200, and 300 trees per hectare. We used 15 of these plots. There were other plots, and we divided as I explained. And steady state was reached since the thinning in 2009. So that's important to say. It's not something that we just thinned the plots and checked. This has been for many years now. And we only selected the years which we had very consistent data collection in. So first in the results is the germination of seedling and their survivorship. And you can see the germination in those gray boxes, which you can see by germinants per hectare numbers, and this is a logarithmic scale. So numbers at the thousands or even tens of thousands, depending on the precipitation per year. But the survivorship, if you see, these are much lower numbers. These are the red boxes down there. So in some years, we had survivorship of more than 8% of these seedling, which is also very low. But other years, especially dry years like 2018 or 19, this went down to 0.05%. So even if you have thousands of germinants, you end up with 1 or 2 seedlings after the dry season. So germination increased along the year when precipitation increased, but without a significant effect of the climatic conditions. And survivorship was correlated to annual precipitation and almost eliminated in dry years, as you could see here. So how about the effects of grazing and stand density on these processes? So you have here the different densities, as you can see in the bottom, from 100 to 300. And the right side here is without the grazers, and the left side is with the grazing. So germination was positively affected by increasing stand density. And this is maybe expected if you have more trees, more cones that produce more seeds. So you have more seedlings that germinate. But was negatively affected by the grazing, which is also could be expected. And grazing effects were not detected in the survivorship step. So we move next with the life of these seedlings to seedling recruitment. As we explained before, for the seed germination, we saw that there is a benefit with stand density, but the negative effect of grazing. With seedling survivorship that was measured in September, we had a bad effect, negative effect of the drought. And if we look at the numbers, these are the complete numbers of everything that we counted. And these are big numbers, as you can see, along many years. So you see the different densities from 300, 200, and 100. And these are the numbers for what germinated, numbers to what survived. And next, we're going to talk about the multi-year seedling growth. So the lucky ones that did survive the dry seasons and the grazers were those multi-year seedlings. And they had quite interesting growth patterns. Before that, I have to tell you, if you go to the forest now, and some pines survived more than 2 years or even more than 10 years, you wouldn't even call them trees because they are twisted, they don't grow nicely. So there is no viable future without planting to this forest. But still, you can see young trees that survived, and it's interesting to learn how they did it and where and so what were the mechanisms. So if you look at the grazing versus grazing exclusion, you can see the multi-year seedling growth was suppressed by the grazing, as you can see here, and I remind you of the different precipitation in the different years, and there were interannual dynamics. So this is quite a complex way to look at that, but I will explain. So we could not actually, it was impossible to go and monitor each and every seedlings, although we don't have many, but they are sparse over large areas of the experiment. What we could do is make a sub-sampling of the area, and then measure the ones that we can find. And then we divided them into groups by their height. This is what you see here on the y-axis. So some were taller, some were still shorter, and we assume that as they grow, of course, they grow taller. And here you can see the different years again, grazing versus exclusion, and starting from very low to the top, I think, is let's see again. Right, so you have the different densities, and the multi-year seedling growth was, again, suppressed by the grazing, and without the grazing, only the right side, you see that high density maintained short and stunted seedlings, while the moderate density had a uniform height distribution. This is this green area, and the low stand density and the higher had a higher number of developed multi-year seedlings. So if you remember before, there were more germinants in the high density, but it was the low density that they could survive better, where they had more light to develop, and they were not outshaded by their parents, basically. Okay. So if we can complete the picture from before, we have shown that only the interaction between stand density and grazing had a negative effect on the multi-year growth of these seedlings. And these numbers are so small that we had to enlarge them and show that for the entire experiment, we only had about 100 in different densities. So we can defined this as like a silent killer of this marginal pine population. This is not something that we can see in our eyes when we visit the forest, but once you look down and see what happens with the seedlings, you understand that this is something that can affect the population in the long term. And we confirmed the first hypothesis. So the recruitment bottleneck shaped by drought risks, risks the future of the entire forest. This is what was shown here. The second hypothesis about thinning and grazing only partially confirmed, because there was no state that could achieve optimal conditions for a sufficient regeneration process. As I told you before, with the different densities, had different effects. So these suggest and support the inevitable forecast of changes in population dynamics and population extirpation. Not very positive. Can this case study be further generalized? So we know that previous studies show successful seedling recruitment in similar Aleppo pine forests. So if you go just 20 more kilometers north, and you can have a bit wetter site, you can already see a lot of regeneration, and there is a multi-age forest. So that's very close, but still very sharp difference. So still multiple factors and their interaction with the ongoing climate change, similar to the Atir forest, could shape forest dynamics worldwide. And with that, I'm going to thank you for listening. Thank the lab members, Ella, of course. She's not with us anymore, but that's all. Yeah.
Thank you, Tamir. Very impressive data analysis.
So the question was about the effect of shading on the germination. So pines are like pioneer species. They need a lot of light, but for the germination per se, we didn't see much effect. So they can germinate even when it's almost dark. It doesn't matter to them. But to grow well, we show that they grow better when they have much more light, when it's less density. Yeah, thank you. Any?
Is there any seedling germination in autumn?
No,
Germination in autumn?
no germination in autumn. Nothing. No. There is, and autumn here can be considered as even the driest part of the year, because as now, yeah, the rains just start in November. In here it could be December even. So autumn is actually even drier than summer. It's less hot, but so we don't have any germination, nothing. No. Yeah, this is different from Spain. Aleppo pine is after Aleppo in Syria, where it doesn't grow at all. In Israel, we call it Jerusalem pine, but in Spain, you have like 1,000,000 times more what we have here of this species. It's
the reason because I made the question because we have two seasons of germination, the spring and many in autumn. And we are realized that if the Aleppo pine germinates in spring, the chances to survival are too lower compared to all the seedlings that are germinated in autumn. Because they arrive to the next dry season.
I'm so happy to hear that. Actually, I didn't know about it, and thank you for mentioning. You started the talk with the mine and then the tropical. So for me, it's a tropical story what you're telling. Yeah.
I was interested to hear you take it next stage to the practice of planting, so better season to plant very often.
Yes, well, I mean, the best season in Spain is normally autumn to take any plantation and things. But in autumn, the start of rain period or more consistent. And it's much better if all the activities are done in autumn. But well, we have also germination in spring.
Right. effect of grazing on soil moisture? Wow, we have data. Let me. No, we didn't see any. You might expect, because the grazers would eat all the annuals that consume the water, but nothing that we could detect. Yeah, so probably is more complex than that. Could be that trees grow roots more in places that are sparser. Yeah, but we don't know yet. That's a good thing to check. Yeah.
Yeah, thank you very much. I was wondering, did you also try to relate the survivorship and the germination success of that kind to not only to total annual rainfall, I guess let's understood that would be, but seasonal patterns of rainfall? And I asked that because I have long-term monitoring sites in South Africa where I look at also recruitment success without looking at individuals, but I look at numbers. And my data suggests that it actually, total annual rainfall is less important than certain seasons, like particularly the end of the drought season, if that, or dry season, if that is particularly dry and not even little rainfall or very hot, then it's more severe than other times of the year where the rainfall might be low. And it might be interesting for you also for future projections.
That's a great question. That's work in progress. We're actually doing it now since we have the data, but not in this, the analysis we didn't treat it, but that's the exact way to go. Thank you. Yeah. Okay.
So, for the last but not least, I would like to invite Dr. Rakefet David-Schwartz. Rakefet is a researcher at the Agriculture Research Organization Volcanic Center of Israel. She studies forestry genetics and molecular research to enhance forest resilience by increasing genetic variation.
So, hi everyone, and thank you for the opportunity. Is it okay? To give this presentation. So I will talk about forestries and genetic resources that can be used in regenerating forests. But before we start, I want to talk a little bit about wild tigers. So this is Panthera tigris. It's an endangered species. Is it okay like that? Okay. So this is Panthera tigris. It's an endangered species, and surveys from 2010 estimated less than 4,000 tigers remained in the wild. And these 4,000 tigers are separated into very small populations around the world. And as this population becomes smaller, so does the genetic pools of this population, of this small population. And as the genetic pools become smaller, so does the genetic diversity of the populations. And we know that a loss of genetic diversity makes species more vulnerable, less resilient, and they have very hard time to adapt to new environments. Actually, the limited gene pool does not allow them to adapt to new environmental changes. So that's why in 2014, this way? Okay, that's why in 2014, scientists from Stanford University called for interbreeding a subpopulation of tigers. They show that mixing population can prevent this species extinction, and that's the efforts that are going on today. So the story of the tiger actually illustrates how important genetic diversity is for adaptation, for resilience, for evolution, and for sustainability of the species. So forestries are not iconic like tigers, but many species are endangered. And the forest diversity is threatened heavily by climate change effects as wildfires and outbreak of insects, and of course drought. And we know that we need to keep high genetic diversity in forestries to keep this forest sustainability. So, this way, this way, this way. How do I do it? Okay, sorry. So in my group, we study natural genetic variation in order to find genetic resources that can be used to implement in afforestation and forest management to keep sustainability of the forest. And we take different approaches, and I'll talk today about the increasing genetic diversity. And when we say increasing genetic diversity, we usually mean by hybridization. So hybridization can be between two different populations of the same species, which is so-called intraspecific hybridization, or it can be even a hybridization between two different species, which is so-called interspecific hybridization. So today I'll talk about that hybridization between two different species. And the two different species that we are focused on are Pinus halepensis and Pinus brutia. So this is the distribution range of these two species. Okay, so Pinus halepensis is in green, and Pinus brutia is in blue. And you see that this is the Mediterranean basin, and Pinus brutia is concentrated in the wet region of this basin. And Pinus halepensis grow in different places. Most of them are kind of dry. And we know that Pinus halepensis is much more adapted to drought than Pinus brutia. So they can make hybrids because they are genetically very closely related to each other, as you see from this phylogenetic tree of pine species. And they both are planted along the Israeli forest side by side, especially since the last century, the 70s of the last centuries. And they are growing very closely together. And people reported in the past that there is a very limited geographical space in Turkey and in Greece that there are overlap, and then they found hybrids. So when we have them planted across the Israeli forest, we hypothesize that we have hybrids, or they make hybrids in the regenerating forest. So, for example, this one, this tree is very suspected. Like we suspected that tree, it was took by Josu G from Yatir forest. It was an exceptional looking tree. It's very big, and you see around it there are many trees, but you hardly see them because this tree dominating the picture. So this was a suspected hybrid. And I want to talk about today, the presentation of today is a part of the PhD work of Nomi Hominer. Nomi is a PhD student that I'm supervising together with Yagil Osem from Volcani Center and Yossi Rayo from the Hebrew University. She belongs to the Hebrew University, and today she works at KKL, and she's here today if you want to hear more about her project. It's very interesting. So her project was led by questions we asked along the way. So first we asked, do we have hybrids in the forest, in the planted forest? Because we know that the seeds came from different places, and maybe some places were already having hybrid. And then we asked, do we have hybrids in the regenerated forest? Because we know that if they closely together, like they overlap geographically now, so they might have hybrids already. And if we do have hybrids, do they have any advantage in drought conditions? And the last question we ask is, if they do survive the drought, what traits make them excel? So Nomi studies these hybrids in Brutia plots, like Pinus brutia plots, only where Pinus brutia is planted. And why is that? Because she found that the productivity of cones of both species does not overlap. Like Pinus halepensis makes female and male cones much earlier than Pinus brutia, And because of that, it can pollinate the residual pollen from Pinus halepensis hits the young female cone of Pinus brutia, but it doesn't go the other way. So you will find hybrids only in Pinus brutia plantations. Okay, so that was the sites that she studied. And to identify those that suspected tree, she has to develop tools to do that. And the tools that we use in the lab is molecular tools, and these are DNA markers. And it was a long story, but I'll just show here shortly. Like she used a transcriptome-based database that we have in the lab, and she was looking for very specific differences of one nucleotide polymorphism. For example, here we have a DNA sequence, which you see at the bottom, and in this position, it has a T, Pinus halepensis has a T, and Pinus brutia has a C. And all Pinus brutia would have C in this position, and halepensis would have T. So she carefully looked for many, many SNPs, potential SNPs above 100,000, and she found that 97 informative SNPs are good to go for the analysis. Now, there are more, of course, but it was enough for us to go with this research. So the first question we asked, do we have hybrids in the planted forest? And then we chose 3 different sites, and we genotyped them using those genetic markers. And we found that there are hybrids, like this is the ratio of the hybrids. It was about 80 trees, mature trees in the planted forest. This is the forest. They are randomly scattered in the plot because it's planted, and when they were planted, nobody knew that there is hybrids in them, so they were like randomly planted in the forest. And when she measured the tree size, she found that the hybrids are much more larger than the surrounding brutia pines. Okay, so this was significant, and of course it gives us the idea that they might have an advantage in growing in the Israeli forest. So the next question was, do we have hybrids in the regenerated forest? And for that, we chose 4 different sites along the precipitation gradient, and we collected samples from 3 different developmental stages of the regenerated forest. The first stage is the seed production, then the seedlings, like the seeds that germinated at the end of the rainy season, which is here. This is the precipitation. And we also analyze how many hybrids are in the survival seedlings after the dry summer. We don't have any rain in the summer. So NOMI collected seeds and seedlings from the same year, as you see here, from the same rainy year, and the survived seedling at the end of the summer. And she genotyped them using these 97 SNP markers, and these are the numbers she got. And you see hybrids at all levels and at all sites. And when we average to make a clear picture and simple to visualize, we find these numbers. So the hybrid, we have like 5% of the seeds are hybrids in average. And this number is increasing when you look at those that germinated. It suggests that they might have advantage in germination process. But this number was more than doubled when we measure these hybrids at the end of the summer. That strongly suggests that these hybrids have advantage surviving the drought conditions. So using 97 molecular markers also allow us to estimate the level or the stage of this hybridization process in the forest. And I will not go into details of this diagram, but we measure 2 different parameters of the molecular analysis of the markers, and we find that there are many hybrids that are not first generation, meaning that this hybridization process is going on for years, and maybe it's because the mature trees that are hybrid, that planted hybrid, as we saw already in the first experiment. So we want to know like why these hybrids excel, how can they do it, how can they survive the harsh winter, the harsh summer. And the seeds that Naomi collected at the beginning of the season, and she germinated them to genotype them. She kept them and grew them in a controlled environment in Volcani Center and followed the growth parameters and measured many parameters. I'll show here only 3 of them. So she measured total weight, and she found that the hybrids grow faster than the Brutia and similar to Pinus halepensis. They also have a better root system, which is similar to Pinus brutia, meaning they invest much more energy in rooting into the soil to look for more water resources. And they have also in between juvenile needles compared to the mature needles. So the juvenile is the first needle that the pine produce. They are kind of grayish. And then after a year, the pine produce mature needles, which are more resistant to drought. But on the other side, on the other hand, the juvenile needles can make more photosynthesis. So the hybrid is between these 2 species. So it does make more photosynthesis than Pinus brutia, for example, and that's why it grows faster. But on the other hand, it has much more mature needles than Pinus halepensis, and that's why it can survive drought better. We also measured the response to drought stress in a very controlled experiment on the laser metric system at the Hebrew University, together with Menachem Ushelion. And we found very interesting results. We found, of course, there are lots of parameters that we measure, but this is a summary of some of the measurements. Here we see like the stomatal conductance, which also reflects the transpiration of the species. And on the x-axis, we have the soil water contents, which reduces over time. So we see that Pinus halepensis transpire much more water, and that means that it also grows faster. But this point, at this point, the stomatal conductance reduces. It means that the species sense the drought and close stomata to prevent water loss. And Pinus halepensis does it much earlier than Pinus brutia in red here. So these are the difference between these 2 species. And we see the hybrid is transpire a little bit more than brutia, meaning that it grows faster than brutia, but it closes stomata much earlier than both species. So that means that it's much more careful when it comes to drought. It closes stomata and it keeps water inside the plant instead of losing it. And to conclude, we found that hybrid have better germination and better survival rates than either of the species. And we found that they do that by combining physiological and growth parameters that makes them better survive the drought. Okay, so they have better root system, they grow faster, and by growing faster, they also can compete the annual plants easily. And they have less energy invested in mature needles, and they have early response to drought. And by that, I want to thank, of course, Naomi for all this data and very interesting PhD, and our lab members, and the funding agency, the KKL-JNF and BARD, and of course for our collaborations that are on the right. So thank you for your attention. Can start? Yes.
Thank you for your talk.
Yes.
I was wondering, you showed a map of the Mukhaka and Gilat and the percent of natural reoccurrence of hybrids.
Yes.
And it seems that the numbers are dropping as you move to the south, but then you show that the hybrid seems to excel in drought conditions. So how do you explain it?
Yeah, I can explain that, I think, because in Mukhaka and Bekoa, the planted, trees were planted many years ago, and there were also, I know that in the past, KKL used to plant a very dense forest. And they were they were doing thinning actions several times in these plots because they are very, I don't remember the age, but it's about 50 years. So they were thinned, these plots were thinned. And of course, when you thin the forest, usually you take out the smallest trees and you keep the biggest trees around. And the Gilat plot was never thinned. So that's my explanation for these results.
Thank you, Rakefet. So for semi-arid afforestation, would you recommend the hybrid?
Oh yeah, I forgot to say that. Yes, so we recommend, and actually it's an ongoing process at KKL now, we recommend using at least for Yatir forest, because that's the connection to your lecture, Tamir. We actually provide some solution for forest regeneration in semi-arid areas. And I think that should be included in Yatir forest regeneration, or at least at some plots that are about to be planted soon, I guess.
How easy is it to produce this?
So we can identify, we know which plot is the best to produce hybrids. And the ratio, if we look again, we have in seeds, if we look at the seed level, we have like Enhod here, this one, for example, has 13%. We can identify even better plots if we would like to go for this approach, and we can grow them in nurseries and then genotype them quickly. We don't have to use the all 100 SNP markers. We can just use few to identify them, and it's even simpler because we have, if we collect them from Butia trees, we know that it's another story, but the pines, transfer the chloroplast from the pollen. And it's easy to, we have a chloroplast marker that we can, if we take from Butia pine seeds, germinate them and genotype, we can use only one marker to find out which were hybridized by Pinus halepensis. So it's easy to identify it in the, the germinated seeds, and then anyway you grow them in a nursery before you go to plant. So it's possible. Yeah. Yes. Okay. Oh, I'm sorry.
How consistent was the drought-tolerant traits from the different populations? Did you have a big difference?
Oh, we didn't check that. You mean this one, the drought response to drought? No, we didn't check for each site. We just took representative of, and we did like a routine experiment. Like we rooted the hybrids, and we have. It's a very expensive experiment to do, so we didn't do for all sites. It's only for a few.
What was the origin of the seed for the experiment?
This one, it's from mature trees that we rooted. We take small branches, we root them, and we have like clones to measure this kind of experiment. We can measure and make statistic by using the same tree in repetitions for for statistic.
But the parent tree that you
Yeah.
the seeds from, where did it come from?
It didn't come from these plots that we measured, from other trees that we identified before. Yeah. On air.
Do you have any predictions on the carry between generations, those traits?
Yeah, that's the next step. Actually, we have some, I know, like it's sustainable. We think that it's much more sustainable to use this approach, like to have hybrids in the forest. we don't have to have them, they are already there, you know. So we just need to be aware to this process and manipulate and manage the forest when we know this, we have this knowledge. And we assume, because I mentioned that Pinus halepensis is much more drought resistant than Pinus brutia, we assume that more genes from Pinus halepensis would introgress into the Pinus brutia genome. And we already have a clue of one marker that is, it comes from a gene. So our markers comes from genes. So we have one suspected gene that might be beneficial for drought survival, but I didn't want to talk about it here. Yeah. Thanks.
One of the things that I think would be also important to look on is the traits that you detect for the resilience species. So which trait do you suggest to evaluate, and if you suggest to evaluate during time, not just for the specific determination?
What do you mean, like trait that be related to drought resistance?
Also trait that related to drought resistance, but also trait related to forest management or the resilience of those trees in the future. So which trait do you suggest to work on?
I think like the best way to go is to find that these are drought resistance. And the way to see it is to plant them in dry environments. Like Yatir forest, we have already planted plots of these plants in Shacharia, which also has less rain, annual rain. And we soon will have some in Yatir forests, and then we will know, that's the bottom line, like who survive, right? So the best way to go is to plant them in dry environments and to see, follow them and see. Survival, I think. Yeah, I think that's the best way. We can do all what you say, but maybe it's not worth it, and just follow the survivals. Yeah. Yeah.
Why do we need to look for the 10% in the field? Is it possible to course?
Okay, it is possible, but I'm not going to do it. It's really tricky. The action itself is not so easy, but it's possible. But beyond that, first of all, I believe in nature, and we need to find the solution in nature. When we do it, we choose maybe we choose the different genotype, the not good genotype to cross together. If it happened in nature in this environment, so I think that's the best. And also, when you pollinate a pine tree, it takes 2 years to get seeds. That's the biological process. We cannot change that, of course. So it's a very laborious, and people do it around the world when they have breeding programs of trees, especially forest trees, but they do it for wood production, fast growth. They don't do it for drought. It's only now that people start talking about drought resistant in breeding programs. So it's a breeding program. What you're talking about, it's a breeding program. Nobody's going to do it in Israel because there is no wood industry that's worth it, I think. It's a big and, but it's easy to go and look for them in the forest. Thank you.